Hello, everyone, and thank you for attending today's webinar, Moving a Complex Protein from Development to Large-Scale Clinical Production, presented by Catalent. I'm Joe Keenan, contributor to Fierce Biotech, and I'll be moderating this webinar. Our speakers today are Dr. David Rebuka, Global Head of R&D Chemical Biology, Catalent Biologics, and Dr. Greg Bleck, Global Head of R&D Biologics, Catalent Biologics. You can read their full bios on the right side of your window. Before we begin, just a few technical notes. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. You can find additional answers to some common technical issues located in the Help button at the bottom of your screen. We will follow the presentation with a Q&A session. Please submit your questions using the Q&A button on the left side of your screen. That pretty much covers it. Okay, we'll go ahead and begin. Greg, please go ahead.
Thank you, Joe, and welcome to everybody. Today we're going to walk you through a program that we've recently completed here for a complex protein where we've initiated cell line development, process development, both upstream and downstream, and then scaled that complex protein to large-scale manufacturing. To talk a little bit about the background of the complex protein and the process that we're attempting to optimize, David Rebuka is going to take over for a few minutes here to walk you through sort of what we call the SMARTag technology.
Well, good afternoon, everyone, and I will just be spending a few minutes to talk through a technology that we developed at our site out in Emeryville, California, which we call the SMARTag process or SMARTag technology. So basically, what we set out to do was to develop a method or protocols where we could site-specifically chemically modify a protein. So we're making bioconjugates or we're doing bioconjugation in a way where we're able to put a small molecule at a predetermined site. So basically what we've done is we've engineered in what we like to call a bioorthogonal chemical handle. And so all that means is that the chemistries that you would use to react to that handle are the opposite of what you would typically use to chemically modify the underlying 20 natural conical amino acids. So we call this a bioorthogonal handle. And so we engineer that into a protein, and then you can do a subsequent site-specific conjugation where you're only chemically reacting at that handle. And at the end, you end up with a bioconjugate that's been modified only in one place. So you end up with a homogeneous product. Now, this is a bioconjugation platform, and so there are many applications one could apply the SMARTag technology to, and here we're just highlighting a couple of them, a handful. So, for example, one might want to conjugate toxins or payloads to a protein. This would sort of encompass the class of antibody drug conjugates that are used for targeting cytotoxins to tissues. But one could also envision site-specifically pegylating a protein or using things like a variety of different oligomers like DNA or RNA. You could conjugate peptides to this tag through the chemistries that we've developed, as well as sort of protein-protein chemical fusions. Or you can envision possibly even making imaging agents, putting fluorophores onto a protein in a site-specific way. And we've done a lot of this work in a variety of different contexts and collaborations, always with the intent of site-specifically modifying a particular protein with a small molecule payload that we're interested in. And so in the context of sort of a high-level overview of our technology, it is a site-specific method, and in the context of ADCs, We're designing conjugates that are efficacious and optimized. We are anticipating or looking forward to improved consistency of a product or an ADC product and better regulatory compliance. One of the things we'll talk about today is how scalable is the process, and over the next half an hour or so, we'll be highlighting data around that particular question. So the technology came out of research on a class of proteins called sulfatases. And so sulfatases are sort of analogous to other proteins that are involved in sort of biomolecule degradation. And so sulfatases are responsible for the desulfation of biomolecules, and shown up in the upper right corner of the slide. What's very interesting and unique about this class of enzymes is in the active site is an amino acid where the side chain is essentially an aldehyde. And this is an amino acid called formoglycine. And so from a chemist's perspective, that side chain becomes or could become your bioorthogonal chemical handle. So you could theoretically do chemistries on the aldehyde and not touch any other chemical functionality found naturally on a protein surface. So when we were looking at sulfatases, we got very excited about this because we anticipated that this could become potentially a chemical handle that we could then leverage for making bioconjugates. Now, it turns out that there's an enzyme that's responsible for the formation of formoglycine, and that's called formoglycine-generating enzyme, or FGE. And this enzyme is what is naturally found in pretty much all living systems that basically forms this amino acid in the active site of sulfatases. So the way FGE works is it recognizes a very specific consensus sequence, an amino acid consensus sequence, where you have a cysteine which is flanked by both a proline and an arginine. And FGE will recognize and bind to this sequence, and then it does a very interesting oxidation where it oxidizes the thiol to generate the aldehyde and create formoglycine from a cysteine residue. And what sort of our big leap in logic was, could we take this amino acid consensus sequence out of its natural context, so out of sulfatases, could we put that into a heterologous protein, and would FGE still recognize the sequence and do that subsequent conversion? And indeed, that is what happens, and that is now the cornerstone for what we call aldehyde tagging, or the SMARTag technology. And so essentially, how we leverage this enzyme is by putting the FGE or the enzyme into a cell line of interest. So in this case, in this particular picture, you can see on the bottom left corner, that's meant to represent a CHO cell. And so you can engineer CHO cells to have this FGE embedded into their cells and stably being expressed. You can produce a protein where you have engineered the tag into the peptide backbone. And as the protein is being expressed, FGE will recognize the consensus sequence, do the conversion of cysteine to formoglycine. The protein then folds and is secreted, and then you just through standard purification methods, you'll be able to isolate a protein. In this case, we're depicting an antibody that's been modified to display the aldehydes on its surface, and then you can come in with chemistries that are selective for that aldehyde, and at the end of the day, you end up with a site-specifically modified bioconjugate or protein. Here we're showing a site-specifically modified antibody-drug conjugate. So again, the process is essentially a simple two-step process. So as you are generating and making your protein, you have the conversion step, where the cysteine gets converted to the formal glycine, followed by a subsequent conjugation step, which is selective to the aldehyde that's been generated and formed on the surface. So we're able to take that tag and clone it into different sequences. So we can program where we want that aldehyde to show up in the context of a protein. And here's a cartoon of an antibody where we're highlighting different regions where we can put that tag into place. And so one could envision making libraries of defined architectures and molecular architectures and then testing them for some sort of desired property and pulling out your optimized conjugate. When we do this sort of screening, we look at a number of things. We want to make sure we're not impacting a panel of biophysical properties that are shown down on the bottom right corner. That includes things like impact on aggregation. For antibodies, we don't want to impact antigen binding nor internalization. Other things like melting temperature, SCRN binding are all specific for the antibodies, but we want to make sure we're not impacting those. And then we do look at immunogenicity. We've looked at we've done a number of different studies to confirm and to give us comfort around the fact that the insertion of this sequence is not going to be immunogenic. We've developed a number of chemistries that are site-specific and react only with that aldehyde, and we won't go into too much detail here today, but just want to highlight that in the end, we are very interested in forming very stable bonds. We don't want our conjugates to degrade unexpectedly, and so we've designed chemistries where we're actually forming a carbon-carbon bond between the payload and the protein of interest. So that's the overview of the technology and how we engineer these modified proteins by leveraging an enzyme which we've engineered into cell lines. And now Greg's going to talk a little bit about the process of protein production.
Thanks, David. So before we go into the details behind the actual program, just wanted to give a little bit of an overview of a number of classes of complex proteins which the SMARTag falls under. So these can include virus-like particles or VLPs, where you need two or more proteins expressed in specific ratios in order to get the correct formation of the VLP produced by the cells. These can include proteins that require cleavage in order to be functionally active, where you need to express an enzyme that cleaves the protein to create the functionally active protein. The one highlighted here is what we're going to talk about today, is the proteins that require additional post-translational processing, and in our case, that's the conversion of that cysteine to the formal glycine by an enzyme FGE. And then finally, numerous different bispecific formats are becoming popular in the industry. These can include two-chain bispecifics, that can include three-chain or four-chain bispecifics, so a number of different formats where you need to express multiple genes inside a cell, and you need those genes, and you need the genes to encode proteins that come together and form the correct structure of that specific bispecific format that you might be looking to optimize. So things that aid cell line development, when you're looking at some of these complex proteins and need to express multiple different proteins in order to get your cell line of interest to produce the correct protein, are things like stability. So you want each of those individual genes to be expressed consistently over the culture of the cells and the development of the cell line. And you want those proteins that those genes are encoding to be expressed consistently throughout the development and culture process. You want the technology to work on multiple different cell lines, because sometimes complex proteins, you might not be able to express them in CHO cells. You might need to look at 293 cells or some other cell format in order to get the best quality protein produced. Also, what can be important is the ability to modulate different ratios of protein expression. And so this can be important, especially if your enzyme that you might need to convert a specific protein or cleave a protein could be have effects on cell growth or deleterious to cell growth. You might need that expressed at much lower levels in order than your actual recombinant protein, in order to get the cells to be happy and full cleavage of your protein or modification of your protein. You want to be able to insert genes very effectively into cells, and if you're adding a bunch of different genes into cells, it'd be preferential if the way you were making those cells didn't rely on a lot of selectable markers. Otherwise, you can end up adding a whole bunch of either cell modifying agents or antibiotics in order to fully select cells that are expressing all the genes of interest that are needed to make that particular recombinant protein. So again, we're in this example we're giving you, we're interested to produce an antibody, so heavy and light chain from an antibody. And then we also need to produce that FGE enzyme, and to get the conversion of the cysteine to the formal glycine, to have basically all those cysteines at that particular site convert to formal glycine, so we can do our specific attachment to the antibody. The process we used here to make the cell lines of interest, we call GPEX. It stands for gene product expression. It utilizes replication and competent retrovectors to insert genes into any mammalian cell line, and we've been practicing this technology upwards of 15 years now, and have a number of products, as you can see, currently in clinical trials. Why we think this has some advantages for complex proteins is that we tend to get high yields with the process, so we get high levels of antibody, in this case, produced. We don't use any traditional antibiotic selection or traditional amplification as part of the process, so our genes are inserted very efficiently by the retrovector, so that can shorten timelines. We don't need to have a bunch of resistance markers in our cell lines and a bunch of different antibiotics in order to know our cell lines are containing multiple different genes. So this allows for the production of complex molecules and the ability to titer the expression of one protein to another relatively easily. And then the technology is extremely genetically, or results in extremely genetically stable cell lines, and so you want that stable expression over time, so things don't change as you're in your 250 liter or 1,000 liter bioreactor for your production run. So initially we took two approaches for the development of cell lines that were expressing antibodies, tagged antibodies with FGE. Initially, the first one was both a pooled expression of FGE and the antibody, so we did no clonal selection. We just introduced the FGE gene, and then behind that introduced the heavy and light chain gene for the antibody to produce a pooled cell line expressing the antibody. And then the second approach was the one that we're moving forward into larger scale development, is where we produced a clonal cell line overexpressing FGE, and then we use that cell line for further antibody production or other recombinant protein production where we need to get that modification of the tag from the cysteine to the formal glycine. So with approach one, we started with our base CHO line. We did what we call transductions, or cycles of transduction with FGE. We did 3 of those, and we ended up with a pooled cell line producing FGE. And then into that cell line, we introduced light chain and then heavy chain, and finally ended up with a pool of cells producing both FGE, light chain, as well as heavy chain that we move forward in the screening process. So with that approach, you can see here, this is analysis of protein concentration versus the integral cell density of the particular culture. This was a shake flask culture. The pool produced about 600 mg per liter of antibody, and you can see the pool is about a specific productivity of about 10 picograms per cell per day. This is about, I'd say, a little below average for us as far as where we're at normally at this stage, but pretty good expression. And the initial antibody produced was 98% converted, meaning that cysteine, 98% of that cysteine has been converted by FGE to the formal glycine amino acid. And so we're pretty excited with this initial data, and did a bunch of work on this protein. But when we went and rethawed this pool and attempted to repeat the process, what we saw was that we didn't get that 98% conversion anymore upon on expansion of those cells, and we saw roughly 50% conversion most of the other times we ran the process. So there was something different between the initial runs we had done with the cell line and the subsequent the runs we did with the cell line that we needed to sort through and see why we weren't getting that full conversion of the cysteine to the formal glycine. And so we did a number of different studies, but one of the ones that sort of helped point us in the right direction was we had noticed when we looked at different media that the percent conversion of the cysteine to formal glycine was different depending on what media that we cultured the cells in. And so if you look at the formal glycine percentage in the CD40 CHO media, you can see early in the culture time we're seeing a little over 80% conversion of the cysteine to the formal glycine on day 5 of culture, but as the culture went on, later and later in culture, you saw down to close to 20% conversion of the cysteine to the formal glycine. In a different media, PF-CHO, we saw relatively high conversion at the start, but then over time we did see some reduction in the amount of conversion we saw on day 14 of culture. So this led us down the path that there was something limiting in the culture media that was important to help the enzyme perform this conversion. And that turned out to be copper. And so you can see here that when you place copper as compared to other heavy metals in combination with FGE, you see a much greater increase in specific activity of the enzyme. So copper seems to be a cofactor that's needed to get maximum production of FGE, and we showed that as part of the development process. So we could take the enzyme, load it with copper, and see much greater activity than if there was no copper present in the enzyme. So we went on and did some again comparisons of media, and you can see in the multiple different medias that we tried, we saw a wide variation in the percent conversion. So I think the take-home message for us is that even though medias are supposed to be relatively consistent in their makeup, even small changes of things like some of the heavy metals, in this case copper, can greatly affect the functionality of the enzyme and subsequently the conversion we were looking to maximize. But in either of the culture media, if we add copper to the media as part of the production process, we see very good conversion of the cysteine to formylglycine. So once we got this sorted out, and so we had that pooled cell line working at optimal levels consistently now, we took the second approach where we actually made a cell line that was over a clonal cell line that was overexpressing FGE, selected different clonal lines, and I'll walk you through how we did that. And then once we had those clonal lines, we did cycles of transduction again with light chain and heavy chain to make our final production cell lines. So we didn't have initially we didn't have a really good assay for FGE, and this was something we were working on developing, and as you'll see in a couple slides that we were able to develop something, but initially we used the criteria that we do around here once in a while if we don't have a good assay, a good high throughput assay for expression of the protein. We'll look at the gene copy number of the cell line, and we use an index we call gene copy index to monitor copy number in our cell lines as we go through development. And so we didn't know if FGE expressed at really high levels might inhibit cell growth, and so we took a variety of different copy number cell lines forward in development. So our initial screen for selection of the cell lines was strictly based on gene copy number, looking for a range of copy numbers in the cell line. Once we had the cell lines narrowed down, luckily we had developed a FACS-based assay where we are able to measure FGE in these cells. The upper left-hand panel is sort of the background noise with a wild-type secondary antibody and the FACS sort. The wild-type cell lines, so CHO cell lines without anything introduced into them, do express FGE, but they express it at a very low level and a level that's not suitable for performing the conversion on a typical manufacturing process, and which is why we had to add the FGE gene to the cells. And so you can see a number of different cell lines out. They expressed at various different levels, the highest one being clone 120, and that's the one we were sort of looking to potentially move forward with based on this result, but did a few other assays I'll describe to confirm that this was truly the best clone to move forward. So subsequently, we developed an ELISA assay that was able to look at the amount of FGE from cell lysates. And again, when we did this analysis, clone 120 was by far the best expressing clone, and again indicated that this was probably the clone that we wanted to move forward with. We also wanted to look at doubling time and confirm that FGE wasn't somehow greatly changing the doubling time from traditional wild-type CHO-F cells, which was our basis. You can see a couple of the clones have very, and clone 120 looked to be a little slower, but it was sort of within the range of error of this particular assay when we did a quick analysis of doubling time. So this was still the clone that we thought we were going to move forward with based on expression, and its growth was fell within sort of normal parameters. So we wanted to make sure the FGE expression was stable over time, and so we looked at all the different clones and cultured them out over 5 weeks of continuing expansion and passaging. And basically all the clones were stable in their FGE expression. Again, clone 120 was stable as we looked to move that one forward potentially. So we took clone 120 since that was our lead after the analysis, and we started putting different antibodies into that cell line and different numbers of tags on antibodies into the cell line to see how well they would perform both from a production as well as a cell growth in various different culture conditions. This initial experiment was done in shake flasks with one particular antibody where we selected the top 5 clones and moved those forward in development. You can see that clone 1 and 2 were both about a little over 5 grams per liter, so we were able to get cell lines that were producing FGE and were capable of sort of traditional antibody levels of production in standard fed batch conditions. And then the bottom graph highlights the level of conversion. So close to 100% conversion of that cysteine to the formal glycine. So the enzyme present in that clonal cell line is capable of keeping up with specific productivities in the case of clone 1 of 75 picograms per cell per day. So there's enough enzyme around to be able to perform that conversion in the endoplasmic reticulum and get the fully tagged protein secreted from the cells. So we're very excited when we saw this data. This just shows the specific productivity curves of the top 2 clones. You can see their integral viable cell densities are pretty good, and they're getting, both clones are showing full what we consider full conversion of the cysteine to the formal glycine. These were 16-day production runs in the shake flasks, and we see normal viable cell densities throughout the culture. So again, we're happy to see that the high expression of the FGE in combination with the high expression of the antibody wasn't affecting cell growth, and we were able to do sort of normal culture processes and hopefully subsequent manufacturing processes with the cell line. We also wanted to tax the machinery of the cell line a little bit more, so we put in an antibody that had 2 tags on the heavy chain. So when you make the tetrameric complex, it has 4 tags attached to the antibody, and so we wanted to confirm that we are able to get good expression and conversion. This antibody, this is a totally different antibody. It doesn't express quite as well, but still we're able to get 1 clone over 2 grams per liter. And both sites, you can see greater than 95% conversion of the cysteine to the formal glycine on the antibody. So now we're looking to scale this up, and so can the process scale up, and can we get this into sort of large-scale clinical manufacturing? So I'm going to give you sort of two case studies. One we're calling antibody 1 and the other antibody 2. So for antibody 1, we scaled up the process we had originally developed in smaller scale shake flask and amber bioreactors. And we took it to 2-liter bioreactors. You can see about 2.5 gram per liter process. It's about 34 picogram per cell per day, specific productivities. And again, as we scaled it into 2-liter bioreactors, we maintained that good conversion. So the formal glycine was still able, the FGE, the formal glycine generating enzyme, was still able to keep up with the quantities of antibody being produced. Viable cell densities for this, then after the 2-liter run, we scaled it up to the 100-liter scale, and these are viable cell densities and percent viability for this 100-liter run. It was a 14-day run. We can see what we typically get are sort of cell viabilities in this range for this particular media and feed strategy, and the runs typically go from 12 to 16 days. So this was right within the parameters we normally see for this sort of media and feed strategy. Antibody yield was almost 3 grams per liter at the 100-liter scale. And again, specific productivity was slightly higher than what we saw at the 2-liter scale, so it transferred very well to 100-liter scale. And then again, the most important thing for us is not only are we getting these high titers, but we're getting that full conversion of the cysteine to the formal glycine. So for antibody 2, again, this antibody is a little lower expressing antibody. So in 2-liter bioreactors, we saw about 2 grams per liter. It's about a 20 picogram per cell per day process. And again, with this particular antibody, it scaled well, and we saw good conversion of the cysteine to the formal glycine. Scaled this up to the 100-liter scale, and cell viabilities were higher than the previous process. We're in a different media and feed strategy for this particular cell line. And this process, because we got to a little higher viabilities, isn't quite as long a process, so it went out for 12 days. So antibody production was right around 2 grams per liter, which was what we had saw in the 2-liter reactors. Specific productivity, again, a little less than 20 picograms per cell per day, and good conversion of the tag. This particular process we have just recently scaled up to the 1,000-liter scale, and we've seen almost identical results to the 100-liter scale. I didn't put that data on here, but it scaled very well to the 1,000-liter scale. And again, the protein quality coming out was exactly as we had observed previously. So conclusions, full FG antibody conversion and good expression was observed using the two different approaches, both the pooled approach as well as the more traditional clonal selection of different FGE expressing cell lines, as well as the subsequent antibody producing cell lines. So high titers and full conversion were observed at all the various different stages of culture. So we didn't see any changes as we scaled up the process. The quality of the protein was the same coming out of the 2 liters as it was coming out of the 1,000 liter. We now have an FGE expressing cell line that we're very happy with, that shows great stability and good expression of FGE, and when you put a TAG protein in, very good conversion of that cysteine to the form of glycine. And so the GPEX process was an effective solution in this particular sort of complex protein challenge. So finally, I'd like to thank a few of the folks that did a lot of this work. Our site in Emeryville, California is led by David, who you heard from earlier, and then our site here in Madison, Wisconsin, where most of the cell line development and process development was performed. So with that, I'll turn it back to Joe, and I think we'll get to some questions.
Thanks, Greg. We'll move on to the Q&A. Just want to remind the audience there's still time to submit questions using the Q&A button at the bottom of your screen. We already got a lot of great questions, and we will try to get to as many as possible. First question up is, do you see higher MAB oxidation or remediation with Cu2 plus spike in media?
Yes, so this is David. I'll take that one. So what we've done is some extended characterization across a couple of different antibodies, primarily using just mass spec peptide mapping, nothing out of the ordinary. And we have seen that there is no difference in post-translational modifications on that peptide backbone, including deamidation and sort of some of the other usual suspects. So it appears that the copper addition to the feeding strategy does not impact those post-translational modifications.
And the levels we're adding are well within ranges that have been performed by a number of different researchers and are even present in a lot of different media.
Why do you transduce the light chain first and separately from heavy chain?
We've seen that antibodies, for a number of different antibodies, putting the 2 chains in independently can yield much better expression. So some antibodies seem to require much more light chain than heavy chain to get efficient secretion and expression and formation of the antibody. And typically the reason we put light chain in first is some heavy chains, or probably a fair number of them, the majority of them can be toxic to cells when expressed by themselves. And so we make sure there's light chain in there. That's not the case for all heavy chains. Some heavy chains are secreted fine from cells, but a number of them can be toxic to cells just because they don't get secreted effectively without a light chain.
Have you tried to optimize promoters used in your GPX retrovectors? Yeah,
we've done early on in development. We've done a number of promoter studies. We sort of settled on one that we've done the most of the work with, but we have looked at other promoters as well as other optimizations of the expression cassette.
How many SMART AGS can be incorporated into an MAB?
So yeah, that's a great question. So Greg showed data where we put 1 tag into the heavy chain to form to get 2 tag sites. We've gone as high as 6, on the heavy chain, sort of the FC backbone. We do not know what the upper limit is, to be honest. We've on other proteins outside of antibodies, we've gone as high as 4 different tags without impacting protein folding or function.
Does the tag affect the ability to use standard purification methods for antibodies?
No. It's possible it could, but the way we've scouted positions that you can put the tag and the antibody, and so we have about 8 favorite sites, and none of those impact the purification using standard methodologies.
Did you see any major differences in the molecule or its behavior as you scaled up?
No, the molecule was, looked the same coming out of the initial shake flask studies as it did out of the 1,000-liter study. So, sort of the same percentage of cysteine to formylglycine, and the as David alluded to earlier, the post-translational modifications were similar between the different scales.
What type of analytics are used to evaluate the SMARTag protein?
Right. So we would use a panel of analytics that would be typical for just any kind of wild type protein to determine its characteristics, aggregation, things like that. We've developed very specific assays and analytics for that to measure the amount of F-gly. So that's kind of the unique functionality that you see on the protein. So in our case, we've developed we use mass spec almost exclusively to determine that conversion. Some of the early methods that we used have been published in it was a Nature Methods paper a few years ago, which I would encourage folks to look up if they're interested in some of the details around that analytics. Since then, we've refined and streamlined some of those methods, but they're all based on a mass spec measurement.
All right. I'd just like to remind the audience that we still have time for questions if you have any more. If not, this looks like it might be our last question. Do you think that each payload actually needs its own linker or attachment site?
Well, yes. So in order to bring a small molecule together with the biologic, you'll need you'll need some sort of chemistry. I know of examples where people are non-covalently sticking proteins to or peptides to protein surfaces, like albumin is a good example. But that's the only one I know of where you don't necessarily need to be thinking about the point of attachment and that chemistry, which would subsequently involve having a linker built into the system.
Do you see any issues with misincorporations when the titers were higher?
No, they, I think the maximum titer that we've gotten to is roughly 6 grams per liter with one of the molecules, and no, it's you see basically the normal conversion of the cysteine to the formylglycine, and there's no significant other modifications that we see that are different than what you would on a normal antibody.
Are there any clinical or preclinical data on immunogenicity assessments that can be shared at a high level?
Sure. At a high level, we've now gone into multi-dose studies in cynos. These would have been tox studies, so we were dosing at pretty high levels, and then we subsequently went fishing for AEA after that after a series of doses, and we saw no sort of anti-drug antibodies being generated. So that's one piece of data we have with cynos. The other immunogenicity data all comes from sort of the cell culture or the cell-based assays that we've used with the Abzena company, where they have the cell-based assays. So that at a high level is what I can share around the immunogenicity. I think we're so far pretty encouraged by what we've seen, and at least one of our molecules is on track for the clinic for next year, which will also provide some more data at that point.
Are you using disposable bioreactors? And if so, did you encounter any issues with extractables and leachables?
Yeah, we are using disposable bioreactors, and we haven't noticed any E&L difference from sort of traditional manufacturing processes. So we're basically doing the same as we would for any traditional antibody, and haven't noticed a E&L profile difference.
What are some of the challenges that you've seen in large-scale production of ADCs, especially random conjugation?
So, I mean, I can only speak to what I sort of been indirectly told and communicated, because all the ADCs we make obviously are the site-specifically modified ones, and we have made small amounts of lysine conjugates or cysteine conjugates as comparators, but we've never moved forward and scaled them. But what will happen in these mixtures is there'll be a subset of molecules, and by molecules I mean ADCs, that will have, let's say, 5, 6, 7, 8 payloads on them, and then there'll be a subset of molecules that have 2, 1, or 0. And both of those subsets of populations bring their own liabilities to the table. In the overconjugation case, you see propensities for aggregation, both in the manufacturing process, but then also subsequently you'll see potential aggregation issues even after dosing, and you'll get sort of a first-pass metabolism in the liver where you'll start to maybe see some hepatotox. And then on the other side, you see a lack of general efficacy if you don't have enough payload embedded onto your molecule. I think if you dive deeper into that, and that was a pretty high level sort of comment, but as you dive deeper into payload placement and where the drugs are conjugating, those dramatically impact things like PK. You get rapid clearance, or you get a shorter exposure time, which is directly related to then the efficacy of your payload. All of these factors come into play when you don't have that kind of control. And so, what we're excited about, and what we and other groups as well are striving to determine is finding these optimal locations, being able to have the control of placement where we get really nice PK profiles, nice long exposure times, and we can start to limit some of the tox issues that come from not having that kind of control. And I mean, at a very high level, that's what we are trying to accomplish here.
Do you also support bioanalysis and immunogenicity analysis for the ADC that you support manufacturing?
Yes. So, well, partly yes. So we do have a big bioanalytics group, and we have capabilities both at our site in Emeryville, in Madison, and then other analytical sites within our network that can support all the way through development and manufacture and subsequent clinical trials for ADCs in the bioanalytical piece. We currently do not support immunogenicity studies.
Can you speak to the degree of conjugation achieved using the SMARTag technology?
Yeah, so in our current manufacturing campaigns, which Greg alluded to, we're now sort of running some GMP campaigns. Essentially, you get complete conjugation. So the conjugation efficiency is greater than 99%. That is in part because we have this bioorthogonal handle, so we can drive chemistries to completion without being concerned about overconjugation or chemistries happening somewhere else. So again, we like the system we're using because it's a simple system. It is a chemical handle that we can manipulate and directly without being worried about off-target chemistries, and so you can really drive things to completion. So that's where we currently are in sort of our more optimized systems where we're moving things to the clinic.
All right, it looks like we're at our final question. Before we get to it, we had a lot of great questions today and couldn't get to them all, but we will be getting back to everyone who submitted personally after the webinar. So our final question today, David and Greg, is do you analyze the DAR ratio after bulk manufacturing at site?
Well, yes. So we do DAR measurements along the way, along the whole process, and again, we're using mass spec methods as well as chromatography methods to determine DAR of different lots. So again, there's nothing special about how we're determining DAR. It's kind of like everybody else.
Okay. I'd like to thank everyone for attending this Fierce Markets webinar and submitting so many great questions. I'd like to thank our speakers for participating and Catalent for presenting today's webinar. The webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.